This video is focusing on lytic replication. This is one of two ways that bacteriophage can make more copies of themselves. Another way that bacteriophage can replicate is through what's known as lysogenic replication, which is covered in another video. Remember what bacteriophage are? These are viruses that use bacteria host cells to make more copies of themselves. And so in this image, what we have here is a bacteriophage, and we have a bacterial host cell with a chromosome shown here in red. And so all viruses need to first attach to a host cell and then enter a host cell. In this case, entry is achieved simply through the viral DNA entering a host cell. The next step in the lytic replication is this viral DNA instructs for some viral enzymes that chew up and destroy the bacterial chromosome. You can see this is no longer an intact circular piece of DNA, but instead fragments of. And so this is equivalent of shredding the bacterial instruction manual and leaving the only intact piece of instruction as the viral DNA, the viral genome. So what's going to happen is this viral genome is going to synthesize viral parts. And so those parts include viral proteins and more viral genome pieces. And so all of these newly assembled viral pieces are the parts necessary to make more bacteriophage. So after assembly has occurred, then it's the synthesis step. And so all of those parts are going to start to self-assemble and turn into bacteriophage. And so you can see these look exactly like the beginning bacteriophage that infected this host cell. And this will continue, and so there'll be a larger number of bacteriophage present until the final step of a bacteriophage lytic cycle, which is the release step. Release occurs when the host cell lyses and all of those newly assembled viral particles, those newly assembled bacteriophage, are released. And so in summary, what we have is a virus called a bacteriophage that's going to infect a bacterial host cell. It does so by attaching and injecting its viral DNA. There are instructions on that viral DNA that will tell the cell to make proteins, and some of these proteins will actually degrade that host cell chromosome, so it's no longer intact. So then the intact viral DNA is going to be used to make viral parts. Those are viral genome pieces and viral proteins. Those different parts are assembled into bacteriophage. So those newly assembled bacteriophage can then be released when that cell is actually lysed. And so each of those newly released bacteriophage can then find another host cell and repeat this process. So we call this a cycle because you can see how this kind of connects in this circular series of steps. And this is known as the lytic replication cycle.